Yes, ma'am.
Okay. As one announcement is there, as the electricity is not there at my place, so I will give you an overview of this lecture, and I will take this class for a few minutes. So you know that I would be teaching you the subject genetic engineering. So today is an introductory class where I will tell you about the techniques and why and how this is used in order to get the desired results that I will discuss with you today. So as we are studying it under the branch of biotechnology, so first we should know what is biotechnology. So it is a branch in which we use methods in that living organism and its parts are used to change or to incorporate a particular character to any uh, another living organism. Or in simple term, we can say that if some particular characteristic we want to enhance that characteristic in a particular organism, so we would take it from the another organism and will incorporate into the second organism. So all these techniques in which a biological organism is used, that will be studied under the branch of the biotechnology. So here we will involve the application of scientific applications to the processing of the materials by the biological agents. So by these biological agents or organism, whatever the material we are getting, either it is DNA, RNA or protein, it must be processed and will then be incorporated into the another organism or in culture medium or in any type of scientific applications where we are going to see the desired results. Now, as we are studying now genetic engineering, so in the genetic engineering, a phenomena which is called as a genetic recombination. Genetic recombination meaning is that there is the exchange of information between the two DNA segments. So when we are dealing with the nucleic acids, there would be the exchange of information in the form of the DNA segments. Means one DNA segment is taken from one organism is has to be incorporated into the DNA of the another organism. So there is the generation of the recombinant, and because the phenomena is involving the deoxyribonucleic acid or nucleic acids, hence this is called as the genetic recombination. Naturally, you have seen that in the cell division process in meiosis, in one phase of the meiosis, genetic recombination, where the exchange of the DNA segments takes place and the recombinants are produced. So this is a common occurrence within the same species, but this genetic recombination can also be produced by the artificial means in the laboratory or in other areas. And this artificial mean will include the transference of a gene of one species to another living organism. So when a gene is incorporated from one species and is transferred to the another living organism, nucleic acid, or we can say the DNA, then such technology is called as the recombinant DNA technology. So in common, this recombinant DNA technology is also known by another name, which is called as the genetic engineering. So the formation of the recombinant DNA and its application, all we are going to study under the branch of the genetic engineering. Why it is called so? Because here we are engineering a genetic material or a DNA or RNA. So these nucleic acids are engineered, hence the branch is called as the genetic engineering. Now, the another thing is what is recombinant DNA? You should know the definition of the recombinant DNA. So the recombinant DNA means is that we are producing a unique type of DNA molecule, and how that unique DNA molecule is produced? It is produced by joining together two or more DNA fragments. So when we are creating a recombinant in which the joining of two or more DNA is to be carried out, which are not normally associated with each other, then that type of DNA would be called as the recombinant DNA. Now, these DNA fragments which we are using for the fusion, they are derived from the different biological sources. Now, what is the recombinant DNA technology? So, the technology in which we are creating a recombinant DNA by employing a series of procedures. So, a series of procedures in which we are recombining the DNA segments which we have obtained from the different organisms, then that branch is called as the, or that technology is called as the recombinant DNA technology. And in this technology, some specific conditions are chosen under which a recombinant DNA molecule is produced and is able to enter a cell. Once this recombinant DNA molecule gets its entry into the recipient cell, then there it will replicate along with the replication of the DNA of the recipient cell. So in this way, we will create the multiple copy of the recombinant DNA molecule because we are interested in the expression of this newly created recombinant DNA molecule. And after the translation process, you will get the desired protein and other enzymes which we want to express in more quantity. Now, the development of molecular biology. So, the molecular biology field in which we study the isolation, incorporation of the DNA and all. So, those molecular biology techniques are generally used in the creation of the recombinant DNA molecule or is used in the recombinant DNA technology or in genetic engineering branch. So, whatever the molecular techniques we are going to study in the discipline molecular biology, that techniques or tools we are going to use in the genetic engineering branch. So the tools which have been developed to enable scientists to examine very specific region of the genomes or genes that are used in the field of the genetic engineering. Now, next thing is that what are the various advances in the molecular biology? So the advances in molecular biology here, the combination of the restriction and modification enzymes. In simple terms, these enzymes are those enzymes which are capable of cutting the DNA at the specific sites. So suppose there is one organism which is a donor organism and the another one is the recipient organism. Donor organism is an organism from which we are taking out some segment of the DNA. So how that DNA is taken out? With the help of an enzyme which are called as the restriction enzymes. So these restriction enzymes, they can cut the DNA at the specific sites from the recipient DNA, from the donor's DNA, and then that specific segment is incorporated into the DNA of the recipient cell. Now, various hybridization techniques along with the restriction enzymes, these techniques enables the application of a wide variety of the procedures. Now, the another thing is that what are the techniques and procedures which we will use in this branch? The gene has to be isolated. For example, DNA and RNA, they have to be isolated if we want them to be incorporated into the another organism. It should be purified, and we can also synthesize a particular gene or DNA of interest. So the techniques and procedure which we will use like gene isolation, purification and synthesis. Sequencing is important because we can say that the strand of the DNA, what are the bases in it, that we will come to know under a branch called as under the procedure called as a sequencing procedure. Now, when we are doing sequencing and all, that will become the part of the genomics in which we will study a particular gene of interest. Proteomics is a branch in which we will see the product of a particular gene, and that is, that after a translation process, a protein would be created. So how that protein would be modified according to our interest and all, that we will study under the branch of the proteomics. Or in simple term, I would say that all these things would be encountered under the branch of the genetic engineering, where we are interested in the desired protein or desired gene expression and all. Now, another procedure or technique which we will use in the genetic engineering is what? Polymerase chain reaction. So in the polymerase chain reaction, a particular DNA of interest is amplified and is cloned in multiple copies. So we can create a multiple, or we can amplify a particular DNA of interest under in the technique called as the polymerase chain reaction that we will study in detail in another lecture. Now, mutagenesis is also called as the reverse genetics. So the mutations, some mutations would be created in a particular gene, and why we are interested in creating a mutation in order to get the expression, required expression, I would say that the creation of a required protein, some mutations may be done in the laboratory in a particular gene. Expression analysis branch or technique would include the transcriptional and transcriptional level. In the transcription, you know that DNA to RNA, so RNA would be produced and then the protein would be created under the process of the translation. So expression analysis would be incorporating or would be including the transcriptional as well as the translational processes. We will use a technique called as a restriction fragment length polymorphism, biochemistry, molecular modeling, and gene therapy. So all these techniques and procedures will be used under the branch of the genetic engineering, or we can say that the recombinant DNA technology. Now, what are the various applications of the genetic engineering? So the applications like quantitative preparation of the biomolecules. So if we want to produce a particular biomolecule, say protein, in a more quantity, then that would be under this term quantitative preparation of a particular biomolecule such as protein. In the creation of the recombinant vaccines, in the antenatal diagnosis of genetic diseases. So before birth, the diagnosis of a particular disease, if it is done, then that would be corrected before the birth. Monoclonal antibodies are created. Cell and tissue culture application also, in this field also, there would be the application of the genetic engineering. And to identify the mutations in the genes, if they are deleterious, they could be deleted with the incorporation or with the inclusion of the genetic engineering techniques and in the xenotransplantation. So those are the various applications of the genetic engineering. There are few examples and there are many. So to detect the activation of the oncogenes, also genetic engineering technique is used. So oncogenes, as I have already taught you, they are the cancer causing genes. So they in genetic engineering branch, they can also be detected. Production of the next generation antibiotics in forensics, in the creation of a biosensors, in the creation of the genetically modified crops, and also in the bioterrorism detection, the genetic engineering would be beneficial. Now here a few examples with explanation are given, like quantitative preparation of biomolecules. So for example, if we have isolated a molecule from the higher organism, the availability will be greatly limited. Means you have obtained some molecule, for example, DNA or RNA or a protein from a particular organism, then that would be in limited quantity. So in the genetic engineering, we want to increase the quantity of that particular material which we have obtained from a particular organism. Because why we are interested in creating in a large quantity? Because we are ultimately going to use it in some application. For example, to get 1 unit of the growth hormone. So particular growth hormone, we want to increase its quantity. Then if we want to get 1 unit of a particular growth hormone, then more than 1,000 pituitaries from the cadavers are required. But this is very, we can say, tedious. So for overcoming this, we will use the recombinant technology process. And in this recombinant DNA technology, that has enabled the large scale availability of a particular material by employing various techniques like PCR and others. So what are the risks of contamination in the recombinant DNA or genetic engineering technology? The risk of contamination is also eliminated, and now it is possible to produce a biological substance without any contamination. Now, hepatitis, for example, is caused by the hepatitis B virus, is highly contagious. And by using this technology, the risk of contamination and the risk of the disease caused by such virus is eliminated. Preparation of vaccines or clotting factors are also free of contaminants such as hepatitis B particles. So we can say that RD technology or recombinant DNA technology, it provides the answer to produce the safe antigens for the vaccine production. Now, specific probes for diagnosis of diseases. So what do we mean by the specific probes? Probes is nothing but a nucleotide chain. So a nucleotide chain or a polynucleotide, a short polynucleotide chain would act as a probe. Okay, so for the specific probe, why we are designing? Because we know that due to a base pairing with the complementary DNA, these probes can easily detect the particular gene which is defected or which is the cause of a particular disease. For example, specific probes are useful for the antenatal diagnosis of genetic diseases. And for example, there are many diseases which are caused due to the defect in a single gene. So if the single gene present in a genome got defected, it may lead to a disease that may be deleterious for an organism. For example, there are few examples which are given like cystic fibrosis and phenylketonuria. So in cystic fibrosis, the symptoms are that there is the production of thick mucus and ultimately the bronchi would be blocked. So that is due to the defect in a particular or in a single gene in the case of the cystic fibrosis. I would say that the regulator, that is cystic fibrosis, transmembrane regulator got defective, and that will lead to the disease caused as a cystic fibrosis. In phenylketonuria, as in this disease, the amino acid phenylalanine is not broken down efficiently by a particular organism because due to the defect in the gene which is the cause for the production of an enzyme for the degradation of the phenylalanine. So if the enzyme is not produced in a sufficient quantity by the defective gene, then what would happen? The amino acid phenylalanine would not be broken down efficiently, and in turn, it will lead to the increase in the concentration of phenylalanine in the particular organism. So the increase in the concentration of phenylalanine due to the absence of its degradation will lead to a disease called as the phenylketonuria. So all these two diseases, as I have given here for an example, are caused due to the defect in the single gene. Okay, so that single gene, defective single gene could be diagnosed using the recombinant DNA, or we can say the genetic engineering technology, in which specific probes are designed that can detect that single gene defect and can correct it. So all these things would come under the antenatal diagnosis of genetic diseases, and it could be identified by taking cell samples from the fetus. Now also the probes can be useful for the diagnosis of the viral particles or the bacterial DNA in the suspected blood and tissue samples. Now next thing is to demonstrate virus integration in the transformed cells. Also the genetic engineering technology would be beneficial to detect the activation of the oncogenes in the cancers, to pinpoint the location of the gene in a chromosome, and also to identify the mutations in the genes. Now, next topic is about the gene therapy. So, gene therapy is an important application of the recombinant DNA technology. And what we do in this gene therapy here, the normal gene can be introduced into the patient suffering from a particular disease, and that genetic disease which is the cause of the particular defect in a gene can be corrected. So a normal gene would be introduced in place of the defective gene, and a patient would be cured. So this will come under the we can say under the branch which is called as the gene therapy. Now, how we will find one gene in the large genome? We know that the genome is very large, and how a single gene would be identified in the large genome. So there are various approaches, and there are three basic approaches for this. First is cell-based molecular cloning. In the cell-based molecular cloning, here the creation, or we can say a production and the isolation of a bacterial strain is done that replicates a copy of your gene. So here a gene is replicated in a prokaryote, which is a bacterial strain, and this would help in the cloning, or we can say in the creation of a large quantity of the gene of interest. Now, polymerase chain reaction, as I have explained before also, in that many copies of a particular or a specific region of DNA can be created. Okay, and that DNA which is now produced in multiple copies can be used for the expression of a desired protein or RNA. Now, hybridization would also help in finding out the gene in a large genome. Here, the DNA is first made single-stranded, and then it is allowed to form a double-strand DNA using a labeled version of your gene to make it easy to detect. So in it, a particular strand of DNA is there, then we will use a primer, and then the complementary DNA or RNA is get attached to the single-stranded DNA. So that will be studied in the hybridization technique. Now, what are the basic principles of the recombinant DNA technology? Or we can say that what are the steps of the RDT? So the first is the DNA is inserted into the another DNA molecule. So from the donor organism, we have taken out the DNA, and then that DNA is inserted into the another vector molecule, and that vector molecule is also a DNA molecule in nature, and that in that vector a particular DNA of interest is inserted first. Then the recombinant vector, why we are calling it as a recombinant vector, because it is now having a foreign DNA in it, so it is recombinant vector, and that recombinant vector is now ready to be introduced into the host cell, where it will replicate itself and the gene is then produced. So first is the insertion of a desired DNA into a vector DNA molecule, and then that vector DNA is introduced into the recipient cell, where we want the replication of the DNA of interest. Now, you can see with the help of this model, the basic principle of recombinant DNA technology. So this is the donor DNA and this is the plasmid DNA. So here the vector which is used is a plasmid DNA. Now a particular site which is specific for the restriction endonuclease. So restriction endonuclease will cleave or will cut the particular 5' and 3' sites on the plasmid DNA molecule. So this, you can see the cut is there in the plasmid DNA. Now this is the donor DNA. So the donor DNA is the DNA from which we will cut the DNA of interest. So from this donor DNA, a particular DNA segment is cut with the help of the restriction endonuclease. So this is the DNA which we have obtained from the donor, and this donor DNA or the desired piece of DNA is now ready to be introduced into the cut portion of the plasmid DNA. So how it is done? It is done with the help of the another enzyme which is called as the ligase. So ligase will fuse this desired DNA piece into the cut portion of the plasmid DNA. So you can see that this red colored portion of the DNA segment which we have got from the donor cell's DNA. So now it is ligated to the plasmid DNA, and hence the molecule which is created is called as the recombinant DNA molecule. Now this recombinant DNA molecule, we will use it to incorporate it into the host cell. Now here you can see a cell which is a host bacterial cell is shown. Now this recombinant DNA, which is in the plasmid vector, is now introduced into the bacterial cell. Once it got its entry into the bacterial cell, it will replicate itself into the multiple copies, and also with the expression, it will produce the proteins under the translation process. So you can see the protein which is encoded by the clone gene. So in this way, the desired protein can be created in large quantity using this recombinant DNA technology. So till here, if any question is there, you can ask me.
No, ma'am.
So all of you are registered, have completed your registration process.